Now that you're done with differential gene expression analysis, we would normally stop the step 5 script at this point, but I really wanted to add something. I try to add something new to each iteration of the course, and there's been some exciting developments since the advent of these pseudo-alignment tools like Callisto and Salmon to be able to look at that transcript level data in more detail and to use it to carry out additional kinds of analyses beyond DGE analysis. In particular, what I referenced in the opening slides, which is differential transcript usage analysis, or DTU. And so what I'd like to do in the last part of this lecture is focus on how we can do this DTU analysis without really leaving R, without going back to remap our data in some other way, without doing anything additional in terms of backtracking. And this is really, I think, one of the big differences in the last few years when we think about this idea of differential transcript usage is it used to require using other kinds of alignment methods that were base aware, like traditional aligners, or using other sort of command line tools outside of R that were computationally intensive. I mean, the tools for differential transcript detection or isoform detection have really been based around some of the methods that were developed to identify exon usage from traditional aligners. And so the approaches have been around for a while, but it's been only recently that we can kind of use them directly within R. And a lot of that comes out as a result of being able to map reads to transcripts using these pseudo-alignment methods. So let's just talk briefly about this. I mean, I told you about our workflows for thinking about differential transcript usage and that it would involve Callisto, but would also involve differential exon sequencing algorithms and approaches encompassed in DexSeq and other tools. But we're not even going to leave R, and you're not even really going to get under the hood of DexSeq to do this. And so I think we all appreciate that isoforms are a big part of the gene expression landscape in complex organisms. And in fact, through large consortium studies like ENCODE, we know that most genes produce multiple isoforms, up to about 6 on average per gene. And multiple of those isoforms that are produced have protein coding potential, about 3 approximately per gene. And we know from many diseases, including cancer, which I'm showing here, that isoforms have a strong relationship or association with disease. When a tumor develops, regardless of the sort of cancer type we're talking about, if you do isoform usage analysis in healthy versus tumor, you can detect many situations, many genes with isoform switching that have functional consequences. And we'll talk about what this means to describe isoform switching in the context of functional consequences in just a minute. Those isoform switching events are happening in oncogenes, and they're found in a substantial fraction of multi-isoform genes. And in many cases, they're associated with a different prognosis. All right, so this is patient survival by time since diagnosis, and if there's no isoform switching, you can see one trend line here for survival versus another if there's an isoform switch or a large isoform switch. All right, and we'll talk about how to relate these isoform switches to functional consequences again in just a minute. But and this is true not just in cancer, but also in other diseases or conditions. But I will say that I think this has been largely underexplored in RNA-seq data analysis. An estimate by the same first author, whose R package is the one we'll use today, Viting Syrup, that they estimate that only about 10% of RNA-seq studies explore isoform usage. And that's not because we aren't interested. I mean, everyone appreciates that there are alternative splicing events that lead to different isoforms emerging from the same locus. There are alternative transcriptional start sites. There are alternative transcriptional termination sites. All of these variations lead to different isoforms being expressed from the same locus. And so the challenge is not that we didn't understand or appreciate that this is happening broadly across the transcriptome. The challenge is, what are the tools we have at our disposal to study them, and what are the hurdles or obstacles, as you've seen, right? How to get the software, how to install it, how to use it, how it compares with other tools you might use, what are the statistical approaches you should use. How do we sort of think about approaching the analysis with those aspects in mind? And so we can think about an approach, or one approach, to defining an isoform switch, which is to look at two isoforms. So we'll take a simple example where one isoform is the complete gene model, let's say, and the other isoform is missing this large exon right in the middle. And if you look at the expression of these isoforms in the condition, in the healthy versus tumor, we can see, in this case, isoform 1 is the dominant isoform, but there's a switch that happens in the tumor. That isoform is much more lowly expressed, and the other isoform, the one lacking this exon in the middle, comes way up. You could, of course, quantify expression of each of those isoforms using a pseudo-aligner like Callisto, because it maps reads to transcripts, not to genes. And you could express that as FPKM or RPKM or TPM. And you could also measure the expression or total output from the gene as a whole. But the other way you can sort of approach it is to represent the data as an isoform fraction, or an IF. And an isoform fraction asks what fraction of the total output from the gene can be accounted for by that isoform. And so in this case, in this just mock example, that would be 30 divided by 150. All right. If one isoform was the only isoform expressed, 100% of that gene's expression, 150 divided by 151, would be 100% would be accounted for by that one isoform. So here we have 20% accounted for by isoform 1 and 80% accounted for by isoform 2. Obviously, when we represent the data like this, the isoform fraction, the sum of the isoform fractions across all the isoforms would equal 1. Under condition 2, that expression has changed, and we have that expression data. The total gene expression hasn't changed. So this is an example of a gene that's not differentially expressed between healthy and tumor. And so this would not come out of the analysis we just did, and that's really critical. This gene would not be detected as differentially expressed, and we would miss this interesting biology that's happening, because we now see an isoform switch event where isoform 1 now makes up 80% of the output from that gene, whereas isoform 2 is only 20%. We can take this isoform 1 and 2 and its isoform fraction under condition 1 and 2, 20 and 80, 80 and 20, and we can carry out a differential isoform fraction analysis and say, okay, this is up by 60%, and this is down by 60%, right? And then you can, of course, carry out statistical tests on those isoform fractions. So that's the idea behind analyzing isoform usage using the kind of transcript level data that we have. And so all of this is packaged inside of an R package called Isoform Switch Analyzer. We don't have time to really dig in. I would encourage you to check out the documentation for this R package. I'll add the link to the bottom of this slide before I post them to the website. It includes a number of high-level functions for DTU analysis. And when I say high-level functions, I mean functions that actually bundle up or wrap together a whole bunch of other functions, even from outside of isoform switching. And so the basic workflow here is that there's some external analysis that happens first. In our case, that external analysis was, aligning or mapping our reads to the transcriptome using Callisto. So we're done with that. We then have to import that data into R. We then have to detect isoform switches. And that's all carried out using this isoform switch analysis part 1. They're kind of bundled together, all these different steps inside this one function. And the reason they don't sort of bundle further here is that there's a stopping point here for another external analysis, and it's one that we won't do in this class just because of the interest of time. And that external analysis involves taking these isoform sequences for the isoform switching events. So in the case of the previous slide, that one gene that had these two isoforms, taking those isoform sequences and actually looking at what protein domains are predicted to be present in those. So you actually extract the FASTA files from any gene or any transcript for which there's evidence for isoform switching. You ask about coding potential, signal peptides, disordered regions in the proteins. And that then lets you interpret putative, predicted functional consequences of an isoform switching event. Now, we will not do this. There are some consequence annotations, if you will, that we get out of this for free, but all these things like protein domains, we just won't do. But you'll hopefully appreciate why they can be useful. Once you have these external analyses carried out, you can then use a second part of the workflow to pull these analyses in. It's not required. You don't have to do this step, but it is useful. And it will pull these analyses in, and that's called isoform switch analysis part 2. And that's where you analyze alternative splicing. You predict the consequences, functional consequences of those switching events. So for example, thinking back to that last slide where that one isoform was missing a whole exon, did that exon have critical protein domains involved in DNA binding, for example? Did the loss of an exon result in the loss of a signal peptide, now preventing that protein from being secreted? Did a loss of a particular region create a more disordered protein, the transcript that's more likely to be degraded? All right. So that's the idea of predicted functional consequences of these isoform switching events. We then do a genome-wide analysis of switching to see globally whether there's themes to how the consequences sort of group for these switching events. Do we have a tendency to lose 5' UTRs across our condition? When we switch from healthy to tumor, there's this loss of particular protein domains. And then we can also analyze individual switching events for single genes and ask for all the isoforms available from this gene, how does switching look? And again, these are genes that oftentimes will not come out of our differential expression analysis because the total output from the gene is unchanged. And so that whole process can actually be bundled together in a single function, which is why we're going to do this in the course. Are there other ways to annotate and think about isoform switching? Yes, but no other ones that I know of that can be so easily integrated with our existing workflow. And I will say the work done to put this R package together and to document it is just fantastic. So it really is a powerful approach. So we will use a single function really once we get our data imported into R in the way we need it. A single function called isoform switch analysis combined that essentially allows us to skip this external analysis and drive right through it onto the second part that I just described. Okay, but I have to issue this warning, which is this: if you try, I'm going to demonstrate for you how this works, but if you try to do this using the step 5 script, it won't work. And the reason it won't work is this isoform switch analyzer package was not really handling ensemble mapped data very well. I reached out to the author of the R package. He was wonderful. He fixed some issues in the software and released a new version of the R package that addressed the issue, and now it works fine. But unfortunately, what that means is that to use that newest version of the R package, you're going to need the newest version of Bioconductor. And to appropriately install it and have it work, you need the new version of R. Unfortunately, during the course of running the class, R has released a new update to version 4, and Bioconductor released a new update as well. I know that sounds like oh, we just went through all this work at the beginning of class to install R and RStudio and Bioconductor, but I actually think this is a good exercise for you to try at this point. I think it would be good for you to try to update R and update Bioconductor, because the reality is if you plan on continuing to use these tools on your computer, you're going to need to do this anyway. One of the things you'll find if you choose to go through these steps so that you can actually run exactly what I'm running is that when you update R to version 4, you're going to lose a lot or all of the R packages you installed in the course thus far. And that may seem like just devastating, but you'll get them back in just moments, just by hitting that install button, setting your repositories, and picking up a few of them will actually pull many of the others in as dependencies. So it's not hard to get your entire library back. Okay, so that's the warning. I don't know what to tell you. I'm sorry, but I'm not sorry that you just have to, accept that part of keeping these tools on your computer is keeping them updated. So it's your choice. You can not update and just watch what I'm doing, or you can update your R and bioconductor just by going back to the class on the tools installation and just going through those steps again. Okay, so that's it for that part. Let's go ahead and go now to actually run these steps. Some of these take a while, as you can imagine, for bundling all these steps together. If you're thinking some of these isoform switch analyses may take a while, you're right. So I'm not going to run through all these, but you should have, or you should, if you choose to, you can install the isoform switch analyzer package and just load that R package. Mine's already loaded. And the first thing that I just want to highlight is this program expects certain column headers in our study design file. And so, our study design, our targets, has just sample, there it is, sample, SRA accession, and group. But it's going to look for a column called sample ID. And it's going to look for another column of the condition or variable we want to test, and it wants that to be called condition. So that's fine. It's not uncommon to encounter R packages that look for and require certain column headers. And then I think it's just useful to remove any additional columns that aren't sample ID and condition. So that would be like SRA accession. So the first thing I'm going to do is just modify this targets file. I'm going to call it targets.mod to rename and name as sample ID the column currently named sample, and to name condition as the column currently named group. And then I'm going to just select, dplyr select, the sample ID and condition column. So we'll run that. We'll produce targets.mod, which now looks like this. Great. So now this will play nice with isoform switch analyzer. Now I'm going to create some file paths just like we did back in step 1. And the reason I'm going to do this again is, remember, we sort of summarized our transcript level data to gene level data with TX import. We're basically going to undo that. We're not going to overwrite the TXI gene, but we are going to use isoform switch analyzer's function for reading in data, import isoform expression. And all it needs, it's actually sort of more simple to run than TX import, all it needs is the paths to your Callisto data. And it will import that data into this TXI trans object. So when I do that, it's identifying which alignment algorithm was used, so it sees that it was Callisto, and it's reading the data in, and it gives us some of the same verbiage that we saw from TXI import. It says, hey, reading in these H5 files would be faster than your TSVs, but I'll read in your TSVs, and it's going to go through and read in all 10. It's going to normalize the values via edgeR, not the counts. It's using edgeR to work on the values of isoform usage. And now I have this TXI trans object, which is, again, a large list. And that list contains abundance, counts, length, and import options. Okay. Now that I have this, I want to just make if you look at the column names of the abundance, here it is, the abundance and the counts, it's stripped off the column names. We can take a look at what those are. Currently, they're isoform ID and an abundance underscore 1 through 10. So I want to leave that first column to be named isoform ID, and I think it's important I don't change that. I feel like that's needed. It's going to look for that column header down below. But I'm going to use my sample labels to populate the rest of the column headers. So I can supply that just as a character vector, and it'll populate both the abundance data, abundance data, and the counts data. So I'll run both of those, and now, of course, this looks more appropriate. Okay, at this point, I would import the data, and I will let you do this on your own should you decide to. It takes a minute or two to run, so it's not really useful for me to do this here in a lecture setting. But it is worth talking about what's happening here. So we're going to import that R data. And we're going to import the counts as a count matrix. So it's going to ask for an isoform count matrix, and we supply it with TXI trans $counts. It's going to ask for the abundance measurements, the TPM measurements. And so we're going to supply that to it as well. And that's in the abundance element of that list. It wants our design matrix, and it calls this a design capital M matrix. It's just our targets file that we modified from above, right? That has healthy and disease here. And then there are some parameters here that may be less obvious or apparent as to what they are. And again, I'm going to encourage you to take a look at the isoform switch analyzer documentation. It goes through these in great detail. But the two key things we need to supply here are the updated or the FASTA file for all the transcripts. This is what we aligned or mapped our data to. And we need to provide it something called a GTF file. And we haven't used a GTF file. We haven't talked about a GTF file. I don't want to go into great detail about it, but it's a text file that has essentially 9 columns to it. It has a name of a sequence, a column called seq name, that's the name of the chromosome, CHR1 or just 1, CHRX or just X, or it could be the name of a scaffold or contig if that part of the genome is not assembled into a chromosome. It'll have the source, so what's the name of the database it came from? Is it Ensemble? Is it, what is that source? It'll have the type of genomic feature, gene, exon, transcript, coding sequence, 5' UTR, you name it. It'll have its start and end position. Usually it has an empty character that's just a point, a period, floating point character. And then it'll have a positive or negative for the strand. And then oftentimes, but not always, it'll have whether it's a 0, 1, or 2 frame, indicating the reading frame by base position. Sometimes that's also a point there because they just that information isn't included, so it's point plus point or point minus point. And then it has an attribute, which is just a semicolon separated list of other info about that feature. So that's a single row. All this information is contained for each of these features. And so you can imagine this text file is huge. In fact, the one that I point to in the code of this code chunk is like 3.8, almost 4 million lines long, because it's got annotation information or positional information for each element in the genome. So every gene, every exon, every transcript, every 5' UTR, 3' UTR, coding sequence, you name it. But this essentially provides a map for all known genome features, which really lets you then associate transcripts with their position and with features like exons and coding sequences and 5' UTRs. It gives you the gene models for all these transcripts, because if you remember when we opened that transcript reference FASTA file, it's just nucleotide sequence. This gives us that additional extensive layer of information. And that information is really critical because it lets us sort of combine these two things to get gene model information about the isoforms. And so this is able to be retrieved from the Ensemble FTP site. It needs to be this GTF. There's a couple different ones. It has to be this one that says, CHR, patch, HAPL, SCAF, GTF. And some of this was just reading the documentation for the R package and reading the Google help site for using it. Once you run that, as I said, we'll do all the analysis in one step, right? We won't do that sort of middle external analysis of like protein domains and all that other stuff. So because of that single step, there's really a lot to unpack there. Too much to really unpack, I think, in this lecture and this end of the lecture. So I just encourage you to check out, and here's the link to the documentation for this R package if you're interested in really understanding this in more detail. So once you have that information read in, you carry out the single isoform switch analysis combined that I indicated on the slide presentation, and it will produce an output which you have to set up the folder in advance on your computer. It just has to be whatever you've called it here. I call it isoform output. But when you run this, it will look for that folder to put all the results in. And so if we take a look at what that outputs, and it takes 5 or 10 minutes, I think, to run. Let's take a look at that. If we go to Dropbox, the class, course data set, Callisto output, isoform output. Here it is. So that running that result, let's pretend we did that as a little bit of the baking show approach I mentioned before. It gives you isoform switching events for disease versus healthy, and it gives you all isoform switching events, right? So there's a lot here. It tells you the gene for which that isoform switching event was identified, and it gives you this, which we'll talk about more in a minute, this isoform switch, it's called a switch plot, that gives you several different panels of information, and it does that for each gene in which an isoform switching event was detected. It also extracts the amino acid sequences in a FASTA file, and you also get some genome-wide switching event information that we really won't go over right now, but whether globally the switching observed in your disease versus healthy has an overrepresentation of multi-exon switching or a gain or loss of a transcriptional start site or a transcriptional termination site, sort of globally analyzing to see whether the switching events are enriched in any particular intron, retention, splicing, alternative start or stop sites, etc. All right, so if I ever, after running that, want to take a look at what my output is in terms of a summary, I can run the extract switch summary and it'll just print off, hey, in your disease versus healthy, there were 1,451 genes for which we detected switching events. I can then use the extract top switches to analyze my list of switched genes, isoform switching events and genes, and pull out the top n numbers sorted by Q values. So when I run this, it'll produce a top list similar to what top table did, that I can scroll and see the gene reference, the gene ID, this is the ensemble gene ID, the gene symbol, the condition 1 and condition 2, and then if I continue to scroll down, it lists that Q value, remember the differential isoform fraction analysis that I talked about in the lecture, and then its rank in the list, right? And whether it had consequences to the switching, right? So I can scroll down through this list. I don't see a lot of the differentially expressed genes, so this is picking up an additional layer of analysis here. But some of these were certainly differentially expressed at the gene level in addition to having isoform switching events. How do we know that? Well, let's take a look. We can use this switch plot analysis tool, and this is part of that isoform switch analyzer package that will take several different ways of viewing isoform switching and output them as a single plot. So I'm going to pick one of the genes, Fc gamma receptor R1b, from this list. It's in the top 50, and I'm going to ask to make a switch plot with that gene where condition 1 is disease and condition 2 is healthy, and you can see it's pulling ggplot themes in here as well. I'm going to make my plot window a little bit bigger, and I will just run that bit of code. So the plot that it's going to produce will incorporate many of the things I showed you in the slides leading up to this section as well. I'm going to go back to the plots section. It'll pop up here in just a second. And each of these genes, whether it's in the top 10 or top 50, all of these were output into that folder that I described to you already, into the disease and healthy folder. So if I don't want to hit the switch plot, I can also just scroll down through and take a look at all of these one by one even. So it's taking a little while to plot this. I already actually have it. Oh, there it is. It's coming. So here are all those gene models, right? A lot of that's coming from these GTF files where we know where each exon and each UTR starts and stops. We know the strand that it's on, right? Which direction it's going. And we know a lot of other information. And again, this is a little taxing on my computer right now for some reason. But I'm going to go back to my slide presentation just to highlight that we have several examples if you just go through here of, for example, differential transcript usage. This is a collagen gene. Remember, this is data from the skin. And this is a gene that has 7 different isoforms annotated. And if I take a look, the way these switch plots work is they show me the gene models. They indicate the transcript models for all the transcripts from that gene. They tell me whether there's an indication in my data of decreased or increased usage. And then I get 3 additional plots at the bottom. One about total gene expression between my 2 conditions. One about isoform expression for each of these 7 isoforms across my conditions. And one about isoform usage. This is that isoform fraction I talked about, okay? So if we walk across this data, I now know the model for each of the transcripts, where the exons are, the 3' and 5' UTRs, the strand it's on. And I know that really there's probably no significant difference in the total output from this COL12A1 gene between disease and healthy. So it likely did not come out of my differential gene expression analysis. And it may not have been differentially expressed. Any one of these transcripts may not have been differentially expressed between disease and healthy. Perhaps this first one, but most of these look pretty similar. So the next question, the one that really is dealt with by this package, this R package, is whether there was differential isoform usage. And so in this case, we're talking about the isoform fraction, and each of these conditions, in this case disease, these 7 isoforms would add up to be 100% of the isoform fraction. This plus that, that, and that. And what I can see is under a healthy condition, the majority of transcript usage is coming from this second transcript, this one, and some from this, let's see, 1, 2, 3, 4, 5th transcript, and the 4th one, right? But in the skin of patients with the disease, the majority of the gene's expression is coming from this isoform. There's an isoform switch. It's coming from this one and this one. So there's really this switch from using isoform 2 and 4 and 5 to using isoform 1 and 3, right? And this last one down here as well seems to be differentially used in a disease state. So it's an example of a gene that's not a DGE, it's not a DTE, but it is a differential transcript usage example. We could take a look at other genes like that Fc gamma receptor one that I was trying to plot just a minute ago. And that's an example of one where there is differential gene expression, there's differential transcript expression, and there's also differential transcript usage. So you can see, again, we have, looks like 7 different isoforms annotated for this gene, a really tremendous differential gene expression. So this gene is robustly turned on in the skin of patients with disease compared to healthy. Same thing with these transcripts would all come up probably in a differential transcript analysis. But you still see some evidence of differential transcript usage. Now, this is an example where I think the DTU analysis is probably not that valuable because I'm not sure I care about differential transcript usage if the transcript is not really expressed in one condition or really a big delta in expression of all the transcripts from the gene in one condition compared to the other. So this is a gene that certainly would come out of a DGE or DTE analysis. And although it's significant for differential transcript usage, I'm not sure I really value the DTU analysis in that case. And I'm not sure I would do a DTU analysis in this way for disease and healthy, because I know there's many genes changing dramatically in skin of patients with the disease versus healthy controls. This is an analysis you might carry out in a comparison that's a little more subtle than disease versus healthy. Okay, so back to RStudio, and it of course finished making that plot, and I could do that for any of the genes that I want. And I just want to show you this because it's really a quick and easy way relative to anything else of just really carrying forward from your differential gene expression analysis to doing differential transcript usage really seamlessly. All right, that's it, and I'll see you soon. Bye.